Hello everyone, welcome back to my channel. I hope you all are doing well and you are preparing pretty well for the DBT-JRF examination. And this is the second part of whatever I have uploaded in the part 1 of the mega session of this crash course. So basically this is the second part of the crash course having the mega session, in which we will discuss some of the important topics along with the what type of question can be asked. So stay tuned and do watch it completely to gain the insights of it, and all the very best for your upcoming exam. Hit the like button if you like my efforts and subscribe my channel if you want more. So I have provided so many flashcards and so many important topics as well as questions that can be asked in this DBT-JRF exam. So you can check out my community post as well as shorts to gain insights of it. And in this session also, I have included some of the topics as well as in the summarized form from the standard books. So you can take screenshot or either you can note it down in your notebook and revise as much as you can, because this is very much important and this will be definitely asked in this upcoming exam. So let's start. A very obvious question that is asked from the genetic engineering portion is the different enzymes. This is from the genetic engineering portion, and the topic which is very much asked is about the different enzymes used in the genetic engineering, like the use of alkaline phosphatase. So basically it is an enzyme responsible for removal of the 5' phosphate group of the DNA molecule and the DNase1 along with the E. coli exonuclease 3 Klenau fragment. I have already provided the flashcard related to the different function of important enzyme in the genetic engineering. So this can be definitely asked that what is the Klenau fragment. So basically it is the larger portion after the protease degradation. So the larger fragment has only 5' to 3' polymerase activity, and it has only 3' to 5' exonuclease activity, and it lacks 5' to 3' exonuclease activity. Why it lacks? Because it is transferred into the shorter fragment by the digestion of the whole protein through the subtelicin. And after that, this Mung-Bean nuclease, which is responsible for the single-stranded DNA and RNA endonuclease, it means it cleaves the single-stranded DNA or RNA. This poly-A polymerase responsible for addition of the AMP from ATP to the 3' end of the mRNA. This reverse transcriptase RNase H, which is very much important, which is responsible for the degradation of the RNA strand, particularly from the DNA-RNA hybrid. This S1 nuclease, this T4 polynucleotide kinase is responsible for the transfer of terminal phosphate from where? From a nucleotide 5' triphosphate to a 5' hydroxyl group of a polynucleotide. Now the T4 DNA polymerase and T7 DNA polymerase, stacked DNA polymerase, which is a thermostable DNA polymerase, and beta agarase 1, which is used to digest agarose. So all these enzymes are very much important. If you are unable to remember all of them, at least remember this Klenau fragment, this Mung-Bean nuclease, RNase H, S1 nuclease, along with this stacked DNA polymerase. At least remember, and also including this alkaline phosphatase, because these are the very much important enzyme and also its function can be asked. Now, apart from the enzymes involved in genetic engineering, this is also an important topic, that is the different carrying capacity of the plasmid. So we know that we require a vehicle that is in the form of different plasmid which carry our gene of interest into the host cell. So plasmid is responsible for plasmid, or different vectors act as a vehicle for the transfer of our gene of interest into the host cell. So every vector has a different capacity of carrying that how much DNA insert or how much our gene of interest can be loaded into that vector. So हर एक vector का अपना एक capacity होता है that it can store or it can only take DNA of certain size, like इतना KB तक का ही DNA वो hold कर सकता है अपने पूरे plasmid में. So this is the list of the different vector in the increasing order. The smallest carrying capacity is of plasmid, that is only 0.1 to 10 kb, and the host cell is E. coli. After that, bacteriophage lambda, it has about 10 to 20 kilobase, and cosmid, and its host is E. coli with a insert capacity of about 35 to 45 kb. Phosmid, it has about the same as cosmid. The bacteriophage P1, it has about 80 to 100 kb. And this BAC stands for bacterial artificial chromosome. It has capacity of about 50 to 300. Bacteriophage derived artificial chromosome which has capacity of about 100 to 300, which is higher than rest of the above what we have studied. Now the YAC, that is yeast artificial chromosome. Its host is yeast and it has a insert capacity of very huge amount, that is 100 to 2000 kb. And after that, human artificial chromosome which has capacity of more than 2000 kb. So if it is asked that which of the vector system has the largest capacity of carrying the DNA, so the largest capacity has HAC. And HAC के बाद अगर पूछा जाए तो या HAC ऑप्शन में नहीं रहेगा तो आंसर will be YAC of capacity 100 to 2000. So you have to remember smallest carrying capacity is of plasmid, that is only 0.1 to 10 kb. After that, bacteriophage and then cosmid. So you can remember it in the sequence provided in the list. So please इसको करके जाइए. This can be definitely asked either in the part 1 of the DBT-JRF exam or part 2. Now the third topic of today's session, that is from biochemistry portion, in which the structural hierarchy in the molecular organization of cells is. So you must be aware that the smallest or the fundamental unit of all living organism is cell. So all those cells which are similar in function are coordinated and to form a tissue, and the similar tissue makes up the organ and the organ makes up the organism. So this is what we have studied up till now, that how an organism is made. So in your DBT-JRF exam or in the CSIR exam, this can be asked in the form of very basic question either in the part 1 or part 2 in the matching form. So the first level, that is the monomeric unit, as per the biochemistry, is the different nucleotides for the polynucleotides, amino acid for the proteins, sugars for the carbohydrate. So the different monomeric units combined accumulate to form the level 2, that is the macromolecules, and the different macromolecules makes up the supramolecular structure, and then the supramolecular complex is assembled to form the cell and its cell organelle. So in your examination, it can be asked that which of the following unit is of level 1 organization, level 2, level 3 or level 4. In the level 1, you have the monomers of different polymers. So this nucleotide is the monomer of what? The polynucleotide. This amino acid is the monomer of proteins. This sugars are the different monomers of the carbohydrate. So this is the level 1 organization which is in the form of monomeric unit. So these monomers are repeated to form the level 2, that is the macromolecules. It means this nucleotide makes up the DNA. This amino acid makes up the protein. These sugars when repeated, like the glucose unit when repeated, it will make cellulose and different carbohydrate. Now all these second level macromolecule will assemble to earn the supramolecular complexes. That is, the DNA will be assembled in the form of chromatin along with the histone protein. Now the different protein and the different lipids makes up the plasma membrane, and then the cellulose is the component of the cell wall. So this is the supramolecular complexes, which is the level 3. Now level 4 is this supramolecular complexes like the chromatin, plasma membrane, the cell wall and the different organelles will makes up the whole cell along with its different organelle. So this is the hierarchy of different level of organization. So this can be asked, you can remember easily because this is a very basic and very easy concept. Now the other topic, that is from amino acid, because this is the very much important topic from the biochemistry. As we all know, there are 20 amino acid, standard amino acid, that in different combination makes up different protein. And how they make up different protein? So the one amino acid is linked with the another amino acid through a covalent bond, that is the peptide linkage. So this can be asked, that how the one amino acid is linked up to the another amino acid, or what is the linkage in between the protein's monomer. So it is the covalent bond in the form of peptide linkage, so that makes up the whole protein structure. So this is the one part. Now what is residue? Many times we refer to the amino acid residue. So when one amino acid links up with the another amino acid, so it linked through the removal of one water molecule. So this is known as the residue. When one amino acid is linked with the another amino acid with the removal of or the elimination of water molecule, then that amino acid will be considered as a residue in that protein structure. So you have to remember the covalent linkage, this covalent linkage or the peptide linkage, and what is the term residue. So residue refers to the amino acid when it is linked with the another amino acid by the removal of water molecule. So this is one important point. And apart from that, we have 20 standard amino acid. Now the general structure of amino acid. So the structure of amino acid is common except the structure of the proline, which is a cyclic amino acid. So every amino acid have a R group or side chain, on the basis of which we decide the polarity or the charge or the uncharged nature, and it has the common carboxyl group, amino group, along with the hydrogen group. So this is the basic structure of amino acid. And also you have to remember that all the amino acid are in L stereoisomer. It means L configuration में ही सारे amino acid होते हैं हमारे protein में. Now the single letter code or the 3-letter code that can be asked in the DBT-JRF exam in the first part, that is the part A. So the amino acid classification, first one is this non-polar aliphatic. So on the basis of the non-polar aliphatic, there are glycine, alanine, proline, valine, leucine, isoleucine and methionine. So you should remember the 3-letter code. 3-letter codes are easy, but you have to also remember the single letter code. So G is for glycine, A is for alanine, P is for proline, V is for valine, leucine is for L, isoleucine I and methionine M. And also I have mentioned the isoelectric point. So you can remember that which of the following amino acid has the lowest pi or either the highest pi. And apart from the aliphatic, we have aromatic R groups like phenylalanine, tyrosine and tryptophan are the 3 amino acids that are aromatic in structure. And phenylalanine has a 3-letter code of PHE, tyrosine TYR and tryptophan. Phenylalanine is represented by F. You have to remember it is not represented by P because P is for proline. So phenylalanine, you can remember phenylalanine from F. Now tyrosine is from Y because T is for threonine, and tryptophan is from W. Now the polar and the uncharged R group. So on the basis of that, we have serine, threonine, cysteine, asparagine and glutamine. So you also have to remember the single letter code, that is S for serine, T for threonine, C for cysteine, N for asparagine because A से alanine होता है, glutamine is for Q. So the extreme amino acid you should remember, like the glutamine, the asparagine, the tryptophan, the tyrosine, the phenylalanine. Now the positively charged R group, on the basis of that 3 amino acid, that is lysine represented by K, histidine H, arginine R. Now negatively charged R group, on the basis of that we have aspartate and glutamate. So aspartate or aspartic acid is represented by capital D and glutamate is represented by E. So you have to remember the single letter code, very much important because this can be asked in the part A portion. Now, apart from the 20 common amino acid, we have some non-standard amino acid that are equally important for different metabolic activities in our body. So we should also know about the exact function of the non-standard amino acid, because this can be asked. So among the uncommon amino acid, we have the 4-hydroxyproline. So one thing you should remember is that these non-standard amino acid are the modification of the common residues, that is the 20 amino acid के modification से ही जो non-standard amino acid है वो बनते हैं. So now you have to remember this 4-hydroxyproline, which is the derivative of proline, the standard amino acid, and apart from that the 5-hydroxylysine, which is derived from lysine as the name suggests. Now this hydroxyproline is the plant cell wall protein. So now this 4-hydroxyproline is found in the plant cell wall protein, and both of these, which of these? This 4-hydroxyproline and this 5-hydroxylysine are the component of collagen, which is a fibrous protein found in our connective tissues. So you should remember that 4-hydroxyproline and the 5-hydroxylysine, its function and also its parent amino acid. Now the 6-N-methyl-lysine, which is again the modification of lysine amino acid, which is a constituent of myosin, and as we all know, myosin is a contractile protein of our muscles. Now the amino acid that is gamma carboxyglutamate, which is found in the blood clotting protein, that is prothrombin. We know the essential role of the prothrombin in blood clotting, and also it is found in certain other proteins that bind calcium as the part of their biological function. So this is the role of the gamma carboxyglutamate. And apart from that, one is desmosine, which is the derivative of or the modification of 4-lysine residue makes up the desmosine, and it is found in the fibrous protein elastin. So you have to remember whatever I just compiled in this single slide, that is this 4-hydroxyproline, this 5-hydroxylysine, N-methyl-lysine, carboxyglutamate and this desmosine. This can be asked in different pattern, like in किन amino acid से derive हुआ है ये भी पूछा जा सकता है, and also its important function. So you have to remember the non-standard amino acid also. So one more important topic that is definitely going to be asked in this exam is the absorption of light by the different molecules, or more specifically the Lambert-Beer law. So we all know that a wide range of biomolecules absorb light at a characteristic wavelength. It means हर biomolecule जो है, every biomolecule absorb light at a specific wavelength. एक स्पेसिफिक वेवलेंथ पे हर बायोमोलिक्यूल क्या करता है, लाइट को एब्जॉर्ब करता है. फॉर एग्जाम्पल ट्रिप्टोफेन इज अ एरोमेटिक अमीनो एसिड रेसिड्यू विच एब्जॉर्ब्स एट tryptophan absorbs light at 280 nanometer. So likewise, many other biomolecules also absorb light. So Lambert and Beer have given a relation that how the light is absorbed and what is transmitted. So there is a relation in between the incident light and the transmitted light. So how it is determined that a given biomolecule has absorbed how much light? So this is done through the measurement of light absorption by an instrument known as the spectrophotometer. So spectrophotometer is an instrument which measures the amount of light absorbed. So what it does is, this is the lamp inside the spectrophotometer. So this is the beam of light. Now a monochromator, what monochromator will do? So the monochromator will equalize the light in a single wavelength. I mean, it acts as a filter, that you want a light of wavelength about 280 nanometer, then it will filter all the wavelength to a single wavelength, that is, it will only pass the light of wavelength 280 nanometer. So the role of monochromator is that it provides the light of a only single wavelength. Now this monochromator has thrown the light of a single wavelength, and this light is referred to as the incident light. So this will be the intensity of the incident light, and this is the cuvette in which you have put your biomolecule whose absorbance you want to detect. This is a sample cuvette in which you have taken the sample or the biomolecule, and this is the light that is transmitted after the absorption. So let's say this is, if you have thrown 10 beams, for example purpose I am just saying, and this light has absorbed the 8th beam. Now how much light will be transmitted? So only 2 light beam will be transmitted after the absorption. So it means this 8th beam is absorbed by the sample. Now there is a relation. Higher the concentration, it means higher the concentration of the biomolecule, it means lower is the transmittance. So you can say that there is an inverse relationship. It means higher the concentration of the biomolecule taken in the cuvette, the more is the absorbance and less is the transmittance. Now इसका उल्टा भी यही होगा. Lower the concentration of the biomolecule, then higher the transmittance. यानी जब concentration ही कम होगा जो हमने अपने cuvette में लिया है, तो light तो वो absorb कर ही नहीं पाएगा, because every biomolecule is capable of absorbing light at a certain wavelength. Now the light that is not absorbed is thrown out, and that is known as the transmitted light. So what does Lambert law says about the absorption of light by a molecule? So basically, as per the Lambert law, the fraction of the incident light absorbed by a solution at a given wavelength, it means the amount of light, incident light absorbed by a solution kept in the cuvette at a given wavelength, is related to the thickness of the absorbing layer. It means thickness, it means the path length of the cuvette having the concentration of the absorbing biomolecule. So these two relationships are combined into the Lambert law. Which two lines? The fraction of the incident light absorbed by a solution at a given wavelength is related to the path length and the concentration of the biomolecule. So together we say that Lambert law. Now the ratio of the incident light, that is the log I naught, which is I naught represents the intensity of incident light upon I is the intensity of the transmitted light, the light after absorption which is radiated back. So this is equal to epsilon CL. So this epsilon is what? Epsilon is the molar extinction coefficient, which is represented in units of liters per mole centimeter, and this C is the concentration of the absorbing species like the biomolecule we have kept in the cuvette, and it is represented as moles per liter, and L is the path length in centimeter. Path length of what? The cuvette. So you have to remember this relationship, this log I naught upon I is equals to epsilon CL. So this log I naught upon I gives the absorbance that is represented as A. So if you have to determine the absorbance, you can determine through the expression of log I naught upon I, and this is designated as A. So I have already told that if the concentration is higher, the transmittance will be lower and maximum is the absorbance. Or in other words, you can say that the absorbance A is directly proportional to the concentration of the solution. We can, in other words, say that the absorbance or A is directly proportional to the concentration of the biomolecule. It means the absorbance will be higher if the concentration of the solution is higher. So this is the important of the Lambert law. Now one more portion that is definitely important is the death by misfolding or the disease known as the Prion diseases. So we all know that to be a completely functional, all protein needs to be folded in a complete ordered manner, and if it is misfolded, it will arise some different function that is not required, or we can say that the prion disease arises due to the misfolding of a protein, and it is the causative agent of number of degenerative brain disease. You have to remember the brain disease. It means the prion is related to the brain disease. Now, the best representative of the brain disease caused by the prion is the mad cow disease, or more commonly the bovine spongiform encephalopathy, this BSE. Now, what can be asked in your exam? So, there may be some examples of different diseases, and you will be asked that list out the disease caused by the prion. So, you have to choose the mad cow disease if it is given in the option, or the bovine spongiform encephalopathy. Encephalopathy refers to the different condition related to the brain. So, basically, prion disease are involved in mammals related to the brain diseases. Now, apart from the BSE, that is bovine spongiform encephalopathy, there are other diseases related to the prion. They are known as the Kuru. It means Kuru is also a prion-based disease, and also one disease abbreviated as CJD. So, this CJD stands for Creutzfeldt-Jakob disease in humans, and also one more common disease which we have heard so many times, the scrapie disease in sheep, which is again prion disease, and another one is chronic wasting disease in deer and elk. So basically, these diseases are collectively referred to as spongiform encephalopathies. Why spongiform encephalopathy? Because they make pores in the brain. So the symptoms include dementia and loss of coordination between the organs of our body. So it means the metabolic activity is highly influenced through this disease. And also the prion-based diseases are very fatal. So now a very important part, that what is the infectious agent of this prion disease. So basically, for the prion disease, the prion protein is responsible. Now what is prion protein? This prion protein is abbreviated as PRP. So basically, this prion stands for proteinaceous infectious only. It means ये जो prion हम लोग पढ़ रहे हैं उसका full अगर हम लोग देखें तो PR for proteinaceous, I for infectious, ON for only, and protein. It means PRP stands for prion protein, and prion का भी जो full form है वो है proteinaceous infectious only. So PRP जो है, it is the responsible agent for causing the disease that is known as the spongiform encephalopathy, and prion protein is the normal constituent of brain tissue in all mammals. So अगर बात करें हम prion protein की, so it is the normal protein of our brain that has certain functions, and its role in the mammalian brain is still not known completely, but it appears to have a molecular signaling function. यानी कि जो PRP प्रोटीन है, नॉर्मल PRP प्रोटीन, उसका जो फंक्शन है, इट इज़ नॉट कम्प्लीटली येट डिस्कवर्ड, बट it is thought to have a molecular signaling function. Now the illness occurs, it means some mutation occur that converts this PRP into, through mutation, this is the normal PRP protein which is represented by PRPC, which is through mutation converted into PRPSC, SC for scrapie, which is a prion disease. So basically, this PRPSC represents the disease form of the protein, and this PRPC is the normal form of the prion protein, that is have a role in molecular signaling function in all the mammalian brain. So if it is asked that which of the following protein is the diseased one, so you have to select PRPSC, because SC for scrapie and this PRPC is the normal protein. So how it converted into this? This is through the mutation of the gene encoding PRP produces a change in the amino acid residue that is believed to make the conversion of this PRPC into PRPSC. So this is all about the prion. Now, I have mentioned all these topics in my previous video also from day 1 to day 26. So, for the detailed study, you can check that video. I will provide the link. So, these are some of the inorganic elements that act as a cofactor for the enzymes. So, this copper is the cofactor of cytochrome oxidase. This ferric or the ferrous is the cofactor of cytochrome oxidase or the catalase or peroxidase. This potassium is for pyruvate kinase. This magnesium is for hexokinase, and manganese is for arginase. Aluminum is for dinitrogenase, which is very much important. Nickel is for urease. Selenium is for glutathione peroxidase, and zinc is for carbonic anhydrase or the ADH. ADH is the alcohol dehydrogenase or carboxypeptidase. So this can be definitely asked in the match the column format or in the individual form. Apart from this can also be asked that which of the following polymer is a heteropolymer or the homopolymer and its repeating unit. So you have to remember the polymer name along with the type of polymer, that whether it is a homopolymer or whether it is a heteropolymer. And after that you have to determine or also remember the glycosidic linkage, that in the starch amylose and amylopectin have different glycosidic linkage. That is, amylose is unbranched or linear structure, therefore it has alpha 1,4 glucosidic linkage. Unlike amylopectin has along with alpha 1,4 alpha 1,6 because it is a branching. So at every branching it has alpha 1,6, and glycogen is a homopolymer and it has alpha 1,4 and also alpha 1,6. Now cellulose is a homopolymer and it has not alpha 1,4, instead beta 1,4 and chitin, dextran. So you have to remember all of these. Now the lipid raft, which can be asked that what is the component of lipid raft. So basically lipid raft is made up of cholesterol along with the sphingolipid. So this was all for today. So I have provided as much as I can for the DBT-JRF examination. So now a quick tip for the DBT exam. So you have to solve the 50 questions for sure in the B part. It means out of the 150 question, you have to surely solve the 50 question. 150 questions जो B part में होते हैं, जल्दी से जल्दी जितना possible हो आप क्या करें, स्कैन करें उन 50 क्वेश्चन को जिनसे आप familiar हैं, because there are so many question and बहुत ज़्यादा time taking होता है. तो करना क्या है आपको, यू ओनली हैव टू रीड सिंगल लाइन, एंड सिंगल लाइन से अगर आप कंफर्म नहीं हो पा रहे हैं तो दो तीन लाइन और पढ़ लीजिए, बट एज फास्ट एज यू कैन, बिकॉज यू हैव टू स्कैन दी 150 क्वेश्चन और उस 150 में से आपको सिर्फ 50 निकालना है. सो जिसमें आप श्योर हो उसी को अटेम्प्ट करें, बिकॉज बहुत सारे ऑप्शंस हैं आपके पास. एक ही क्वेश्चन में मत लगे रहिए कि नहीं मुझे ये करना ही है. यू हैव सो मेनी चॉइसेस, सो पिक द करेक्ट 50. अगर करेक्ट आप 50 करेंगे सो यू विल फ्रॉम बी सेक्शन 50 into 3 150 मार्क्स आपके ऐसे ही आ जाएंगे. नाउ इन द ए पार्ट, सो ए पार्ट में आपको 50 करना है, बट इन 50 आप वही अटेम्प्ट कीजिए बिकॉज उसमें ज़्यादा आपके पास चॉइसेस नहीं रहेंगे. सो आपसे जो क्वेश्चंस बनते हैं आप वही कीजिए एंड नेगेटिव मार्किंग का रिस्क मत लीजिए. एंड थैंक यू सो मच फॉर वॉचिंग. ऑल द वेरी बेस्ट.